Hi, my name is Breck Baldwin, and I am one of the collaborators on the BioNotate Autism Annotation project. And I'm going to go through how to do some annotations following the instructions. We're starting with the home page, which you should be familiar with, and we're going to go straight to the annotation link, which is here, which will take you to the snippet annotation interface. At the top, you see there's a home link, there's instructions, so you always have access to them. Please read them before you do any annotations. Access to Gene Search, which is a site where you can get a lot of useful information about particular genes. And there's a link to the Wikipedia, which will give you access to information about genes and proteins. And it's a very useful resource in doing this task. And finally, an email link that you can contact us with. Down below, you see under the heading Snippet, the actual text that we want you to classify into whether the snippet is talking about a gene or a protein or not. It's followed by a title, which is generally a good place to get a feel for whether the article is appropriate at all or not. And then below that is the body of the abstract for this article. We don't have full articles, it's just the abstracts that are submitted to the National Library of Medicine. Over here on the right hand side, we have the interface, which is what you're going to be telling us. So the question is this snippet about a gene? And you get to say yes, no, or you're not sure, and then you submit. That's actually quite simple. You can do this right now anonymously without signing in. So you can say, well, I'm not sure, so we'll go on. And that means that there will be an anonymous user that we're accumulating annotations over. You can practice a little bit or get a feel for what's going on. But you won't be identified with you. And also, if you come back, we won't know who you are, so you may end up re-annotating the same data again and again. So let's go ahead and sign in. And just use your name or whatever you're comfortable with. So I'm Breck Baldwin, so I'll sign in as that. And I'm in. So the question now is, I want to look at the citation and get a sense of whether there is anything in here or the snippet that is indicative of mentioning a gene or a protein. The title in this case is Clinical Significance of TT Virus Infection in Patients with Chronic Hepatitis C. This may be have some genetics information or not. I kind of doubt it, but that's just given my experience, but we'll just take a quick look. There's nothing obviously in the title. So I'm just going to scan really quickly, looking for just sort of text that supports the idea of there being particular information about proteins or genes in here. So up here I've read sort of, this is what the article is about, and then we're drifting into this methods section down below. And I'm really more interested in sort of what the objective of the experiment is. And it's pretty clearly it's about hepatitis C, involves the liver. It's not going to really be anything associated with the details of human genetics. And I'll look down a little further in the methods section here. Whoops. And what do we get? It is telling us about who it is, population, then we have our results. Yeah, I'm pretty comfortable saying that this does not talk about a gene. This is just a general article about hepatitis. It's not the kind of thing we're interested in. So is this snippet about a gene or a protein? No. So we submit. The next snippet is here, and the title is 3H keratinosin binding. And this is a pretty complicated looking title, but I see this 5-HT2A receptors, and receptors can quite often be receptors to particular proteins. So I'm going to see if I can find a resource on the web that tells me what this 5-HT2A thing is. I'm hoping it's a gene. The best place to find out information about genes is go to Gene. And we have a link here for you. It should pop up its own window or its own tab, depending on how your web browser is configured. And what we'll do is we want to copy this. Oops. And then we will go and we will paste it in the search bar here. And that looks like I've got a very good hit. So this is 5-H T2A, which is an exact other alias for this particular gene. This is the serotonin receptor. And this is for it in humans. This is another species. We don't care what the species is. We'll take anything. And so I'm going to go ahead and say that this is indeed a gene. So I'm going to say yes. So, and off we go. The next snippet. Our title is Septamer Element Binding Proteins in a Neuronal and Glial Differentiation. Well, that looks promising. They say protein. So differentiation of progenitors into neurons and glia is regulated by interaction between regulatory DNA elements of neuron and glial specific genes. So I'm looking for something a little more specific than just sort of mention of genes. I want to see a gene or a protein being talked about, a particular one, not just the general class of them. And I'm just going to read through and see if anything looks promising. Etc. Genes. Oh, these are all genes. So tyrosine hydroxylase, that is probably going to be a gene. So I'm just going to copy it and take a look up at front in Entree Gene is where I'm going to search. And that link takes you to the gene part of the database. I paste in here the name, and what do we get? Yep, there it is, TH, official symbol TH, tyrosine hydroxylase. That's for people. And there we have it. That's a gene. That's a classic. We definitely think that it mentions a gene. So that's a yes. Okay, here we have a snippet. Does not look promising at all. The title is, ClickHaler, a novel dry powder inhaler, provides similar bronchodilation to pressurized metered dose inhaler, even at low flow rates. This is extremely unlikely to mention anything we care about. It's really about an inhaler. But we'll just give it a quick look. There may be something in there, but I'm going to read it pretty quickly. Pressurized meter dose inhale, blah, blah, blah. Placebo controlled, fine. Maximum expiratory. Yeah, this is just not, this is not what we want, so we're going to give that a no.